Speaker is coming to us from India, and it's 3:30 in the morning, so I'm hoping that Mr. Kumar is indeed still awake and with us. Our next speaker is Manish Kumar, who is an environmental consultant with International Water Management Institute in Delhi. Mr. Kumar's research also provides important groundwork for the proposal that we'll be discussing shortly with our panel members. And he's going to be telling us about plant-water relations and co-occurring high-altitude rhododendron species in Eastern Himalaya. So as soon as you're ready to begin, Mr. Kumar, you may share your screen.
Okay, thank you a lot for giving me the opportunity. And yes, it's 3:30, but 3:10 almost here. I hope you can see my screen. I was kind of half listening to the other presentations that were going on because I was sorry, not meant to see that. A lot of this. Okay, wait, where is my presentation? Okay, yes, you meant to see this. Yes, because I was trying to figure out where do I fit in this whole scheme of things, and this bewildering set of research that I was looking at, including genetics and all that. I'm taking slightly backseat and taking it at a slightly broader level, looking at how plant-water relations uh, happen or are characterized in rhododendrons in Eastern Himalayas. I'm a PhD student, like my thesis is under review with ATRI, which is a research organization in India, and I'm also a consultant with the International Water Management Institute. My PhD work has been in Eastern Himalayas, which is part of which is a part of the which is eastern part of the Himalayan setup, much more tropical and wetter. And the key aspect of like I've looked at ecohydrology, where we are trying to look at how forests use water, and a key component of that is plant-water relations. What do you mean essentially by plant-water relations? Is generally the process of transpiration that we are concerned about, how a plant uses water from the soil up and releases in the atmosphere. It is primarily driven by the process of photosynthesis, which means that solar radiation and vapor pressure deficit. Vapor pressure deficit is basically an index of dryness. Solar radiation is, of course, the source of energy. Both of them, solar radiation is critical for photosynthesis, whereas vapor pressure deficit kind of allows how much moisture is there in the air, and by that, by a function of it, how much water is pulled out from the tree once the stoma opens for photosynthesis. So it kind of acts as a pump, which is called the transpiration pull. It is also driven by how much nutrient is there in the soil, which is again a main factor for photosynthesis, and how much soil moisture is there, because the moisture is coming from the soil, and that way precipitation becomes a critical factor. And then a combination of these variables with the plant tend to lead to evolution of plant physiological traits over a long period of time. These traits govern how a particular species behaves to all of these environmental stimuli. And in these variables, like vapor pressure deficit, precipitation, or solar radiations, are driver variables, whereas nutrients and soil moistures are generally limiting variables. When two species co-occur, their interactions with these environment can be different based on what sort of traits they have, what sort of evolutionary history they may have, and that makes for fascinating research. The primary variable that we are measuring here is sap flow, which is the amount of water that is being moved from trunk or moved in the trunk through the xylem. And the question that you can ask here in this sort of frame is how plant water use is driven by competitive evolution and environment. How two or more species interact together in a set of environment and either partition water or compete for it in different ways. In turn, they also affect local and regional hydrology, because they are moving a huge volume of water from soil to the atmosphere, and that water can then precipitate either locally or regionally or transnationally as well. Amazon is a classical example where a lot of moisture that is pumped up falls on the other side. And the key focus or the framework of study here is soil plant atmosphere continuum. It's a branch of ecohydrology that has been quite in focus, where people are trying to look at how vegetation interacts with atmosphere and soil in a continuum way. Uh, as of now, there is only one study using Saplow, direct Saplow based method, which are methods through which we can measure how much water is being used by a tree directly. There's only one study from Himalaya, which is the Central Himalaya, Gimre et al. And apart from that, as much as I believe that probably our work in the Eastern Himalaya is the second work that is there. We are still trying to publish it. In this presentation, I'm trying to present the work that we did in a protected area called Kyongnosla Alpine Sanctuary. So this is the whole breadth of Himalayas, and we are located here in the Eastern Himalaya, the state of Sikkim in India. And in the eastern part of Sikkim, we worked in a protected area called Kyongnosla Alpine Sanctuary, which is a range which has an elevation range of 3,600 meters to as high as up to 4,700 meters. 4,700 meters is the range of the watershed. 4,200 meter is the range of the sanctuary. It shares boundary with China, and it is dominated by subalpine conifer forests in the lower region, and then rhododendron scrub as you move above the tree line, and then there are snow dominated no forest or no vegetation area. The precipitation is pretty high in the Eastern Himalayas. It can go up to 4,900 mm, out of which roughly 10% can be snowfall, or 10% or more. Actually, there are very few measurements on snow from this region. So it is quite wet. It is high altitude in the sense that these are above 3,000 meters. So this snow plays a major role in this landscape. The questions that we are trying to address here are what is the plant water use pattern vary between rhododendrons and co-occurring rhododendrons and conifers? What are the functional traits that drive these water use strategies in co-occurring rhododendrons, and how climate change can impact? And this is something that is for the future, but I probably can comment upon it a bit here. It's out of the scope of the study. This is the functional trait part of key species in Sikkim Himalaya comes from the work of Dr. Shweta Basnet, who is a co-collaborator with us, and her PhD was also in the same landscape from A3 only, and she looked at she basically looked at rhododendron distribution from low elevation to high elevation, and how all species, like Sikkim has something like 36 species of rhododendron, or 36 or more. I'm slightly confused on the number. I am taking out the two species that I was interested in, which is Campanulatum and Thompsoni. And as you can see that the distribution of the upper panel is girth distribution, size distribution, and the lower panel is number of individuals of rhododendrons. And the black are Campanulatum and the blue are Thompsoni. And as you can see, the Thompsoni are basically a higher in number at the lower elevation. Their numbers decline from low to high elevation, whereas the number of Campanulatum increase from low to high elevation. So Campanulatum are mostly high elevation species that is a bit distributed in the low lying areas. Low here is 3,600 meters, whereas Campanulatum is a more densely located in the lower elevations. The girth classes are even for Campanulatum, whereas Thompsoni has more wider or larger individuals at mid elevation or its upper range actually. If you look at the leaf functional traits, so this is, if you can see, the red is Thompsoni and the purplish flowers are Campanulatum. So you can see the Campanulatum is more like a shrubby tree, whereas Thompsoni can be slightly larger individuals, and both co-occur at this photograph is around 3,600 meters. The leaf thickness wise, there's not much difference, although Campanulatum has longer leaves and longer and leaner leaves, whereas Thompsoni has more broader leaves. Thompsoni also has larger surface area. They have slightly similar leafing duration and flowering duration as well. So here is the leaf differences. As you can see, Campanulatum here slightly more elongated leaves, whereas Thompsoni has more broader leaves. And this is Shweta here doing field work. Again, Campanulatum here and Thompsoni here. The size difference can be seen quite nicely. My work was looking at how flow or plant water use varies between these species. So I instrumented three species that were there present there: Rhododendron campanulatum, Thompsoni, and Abies densa, which is the dominating conifer. The number of trees varied from 3 to 2 to 4, and their DBH and sapwood length was more or less similar. We didn't find evidences of any hardwood formation in both campanulatum and Thompsoni, whereas densa you could find the evidence of sapwood formation. This is kind of the pictures of the Saplow. So these are the Saplow sensor. These sensors I made by hand. When you buy them, they can cost you around 900 euros per sensor. And for me, it costed around roughly 8 to 10 euros, I'll say. Although a year of my PhD time, a year and a half maybe. We attached them in the trees at different sapwood depths, ranging from outer to inner xylem. And so this is the rhododendrons. This is a conifer. We attached solar panels, and these are a lot of batteries and a data logger sitting there. And at the end of a whole winter, probably 8 months of data collection, I got roughly 8 days of data out of this. I did it a previous year in a temperate forest, and there I had good amount of data, and that I used for my chapter. This was, we somehow lost a lot of data just because the solar panels didn't function. This was a high altitude area, the batteries didn't function. So what I'm presenting is a small subset of almost a 8-day worth data, but it throws up interesting questions. So this is a subset of like roughly 6 days of data. What you see on x-axis is sap flux density, which is the amount of water moving through a per unit area of the trunk. And the three colors are: red is conifer densa, Abies densa. The blue is Thompsoni, blue is Thompsoni, and the green is campanulatum. And as you can see, one first thing that you see is that Abies has slightly wider timings. So the conifer, which is a much larger individual, has slightly wider timings that it transpires from longer during the day than the rhododendrons. In the rhododendrons, Thompsoni has definitely like much higher sap flux density per unit area or sap flow per unit area than campanulatum. Campanulatum had very little sap flow that was occurring, and there's like the day-to-day variability was not very high. When you take an average of these for a day, so x-axis has basically day hours, which is 0 hours to 2300 hours. Then we can see that Thompsoni has the highest sap flux density and campanulatum has the lowest. In all three, the onset is around 7 o'clock in the day, and it continues up to 4 o'clock in the evening. When we multiplied it by the total sapwood that was present in the tree trunk, which then gives you the volumetric sap flow, how much total volume of water is being pumped. The volumetric sap flow was like magnitudes higher in campanulatum, in Thompsoni than campanulatum, and Abies was midway. We found that campanulatum was transpiring at very, very low rates in these systems. We looked at the drivers of these transpiration. So we have the three species in the upper panel, and on x-axis we have, so the first panel is volumetric sap flow, then you have vapor pressure deficit, then solar radiation, and these two are soil moisture sensors that we had placed, and all these are basically for a day, like averaged for a day. So x-axis you have day hours, 0 hours to 2300 hours. And as you can see, sap flux basically started with basically vapor pressure deficit and solar radiation. Both were kind of drivers of onset of sap flow. And but in the midday, the vapor pressure deficit dropped very drastically. So it seemed that it might be primarily driven by solar radiation in that period of time. And it did cause some amount of decline in the soil moisture, at least in one of the sensors, which means that the water that is being taken up by the trees from the soil is reflecting in a diurnal decline in soil moisture in a day. When we did a simple regression model, or just this is a scatter plot. We did a regression model also here. What we found was that for abyss densa solar radiation, like I'm not going to actually densa more. I'll focus more on Campanulatum and Thomsoni, which is the lower two panels. And we have first vapor pressure deficit, then solar radiation, and then soil moisture. What we find is that that there are certain time of the days in which the high vapor pressure deficit is an integral part for driving the sap flow. Solar radiation it has a better result than vapor pressure deficit. So there are periods of high VPD and high sap flow. There are also periods of high VPD and low sap flow. And with net radiation or solar radiation, it has a much better relationship. But there's a lot of hysteresis in the system. Soil moisture doesn't so much relationship. So to tease it apart, I did a 3D plot on which we have on the x-axis net radiation, y-axis vapor pressure deficit, the two drivers. On z-axis, we have the dependent variable, which is the volumetric sap flow. And we have three colors, which is red is densa. Sorry, the Campanulatum is green, and Thomsonite should be blue here. So what we find here is that most of the values of Thomsonite are very high, are more towards the axis of VPD, which means that they are being more driven by the atmospheric dryness, whereas the values of Campanulatum, which is green, are lying more closer to the axis of net radiations. So it seems to suggest that the transpiration in or sap flow in Campanulatum is driven by the availability of sunlight, and whereas in Thomsonite is more driven by the index of atmospheric dryness. Densa was midway distributed, so it seems to be affected by both. So what we conclude from that is one, that Campanulatum seems to be a conservative generalist, which has a very low water use, so it is saving a lot of water. It is driven by the availability of sunlight, and thus vapor pressure deficit or moisture, because it is using very low amount of water, vapor pressure deficit is not a big problem for it. It's the availability of sunlight which is a problem. For Thomsonite is opposite. It has a very high water demand. It's pumping a lot of water out from the system, and thus the transpiration is only limited when the atmospheric dryness is too high. So basically, it is at a threshold level. It basically can't pump more water. Both co-occur, but if you remember, as we see the distribution, the Thomsonite is more dominated in the lower areas, whereas Campanulatum is more dominated in the higher area. So you can postulate a theory here that Campanulatum might be a species that has migrated from high elevation to low elevation. And as we know that at high elevation, sunlight or temperature can have much higher control on a species plant water use, and thus has in generally quite low water use, even if it is present in low elevation. Whereas Thomsonite has migrated from low elevation to high elevation, and it behaves like a low elevation species, which has plenty amount of sunlight. So it is only limited by amount of moisture that is present in the system. So with climate change, if there is a decline in precipitation, and it's going to and with decline in precipitation, which means the moisture is going to decline and is going to become more warmer, which means that VPD might shoot up as well. Thomsonite is more likely to be impacted by it than Campanulatum. Again, this is all theoretical. It's very limited data, but it does put up on that question. Densa kind of treats the middle path, where it is driven both interaction, and it's driven by the interaction between net radiation and VPD, abyss densa being the conifer here. So to summarize, I think it will be interesting to see, again, this was limited to a few trees at a one elevation, but we hope that we can do similar setups across elevation and see if the idea of certain species acting as as a source of very conservatively, whereas certain species being extravagant in their water use can be more concretely established. We also want to postulate this idea of rhododendrons as a keystone species in Indian Himalayas, because they do seem to play a key role in supporting a lot of flora and fauna in the system, and they have other diverse uses as well. This is my acknowledgement. My supervisors feel researcher field support and the fundings that we got. Thank you. I will be happy to take questions if we have time.
Thank you. Excellent talk. And thank you for getting up in the middle of the night to give us that great talk. We have time for one question. We do need to move on to our panel. But if anybody has a question for Manish in the short term, I will.
Can I do a quick one?
Yes, please. Go ahead.
So I'm obviously curious about shifting climates and how you might expect rhododendrons to handle greater droughts, greater flooding. How do you think that rhododendrons in general might handle those two extremes, and or those two extremes simultaneously in the same season?
I just lost you for the two extremes part. Can you repeat the two extremes?
Like flooding and drought. What if a rhododendron experiences flooding and drought in the same season or the same year?
Okay.
Just from a physiological perspective, how can they handle that?
I'll translate flooding and drought in terms of low and high soil moisture conditions.
Yeah.
Because in the mountainous areas we wouldn't have exactly flooding. These regions also don't see drought; they see seasonal drought. And in that sense, the climate change predictions are there that precipitation is likely to decline. Precipitation is likely to go up, but more intensified. As we can see, to some extent, there are evidences that certain species are using a lot more water and have more extravagant water strategies. And if there is more drought condition, which means that precipitation declines, it will lead to, there is also a elevational effect here, in that moisture is generally higher at low elevation and much lower at higher elevation. So if there is overall reduction in moisture, the effect will be higher in the upper elevations, which means that such species which use a lot of water may further contract their distribution. Something like Thompsoni might actually reduce its upward range, whereas something like a conservative might expand because it can still do quite well in low soil moisture conditions. In very high soil moisture conditions, which is that if the precipitation is going to go up, Thompsoni will be something that will benefit from it and might actually move up. And then might drive out, let's say, Campanulatum in competition in those regions. So the effect can be like different for different type of species, or the species with different functional traits or water use strategies.
Great question. This sort of brings us to the next part of